Okay, thank you so much for the introduction, Abhinav, and thank you a lot for the opportunity to also share a bit of our work here and to talk a bit about this I think really interesting field of research. So in the last two lectures, you already looked a lot into very in-depth on how neurogenic lower urinary tract dysfunction presents clinically, how it can be diagnosed and investigated, and how it is currently managed. And then today we will go a little bit more into the look a bit more at the biology underneath all of that. And I'm trying to cover the full system, so from bladder to brain. Just a very short and very simple start, which is likely redundant, but it's just good to keep in mind. So the lower urinary tract or bladder has two functions: storing urine and voiding. And to facilitate that, the reservoir, the bladder itself, and the outlet region have, of course, opposing functions. So during storage, the bladder needs to be relaxed and form like a reservoir for the urine. And the outlet region needs to be closed to prevent involuntary leakage of urine. And during voiding, those functions are switched. So the outlet region is relaxed, opens, and the bladder contracts to expel all the urine from the bladder. And to facilitate that, the bladder wall is made up out of different tissues, and from inside to outside. I think you can see my mouse, right? Yeah. So the innermost layer of the bladder is the urothelium. Then that has a barrier function that prevents urine from penetrating the underlying tissues, and it has an important signaling tissue function. Underneath that is the lamina propria, and that layer contains quite a lot of nerve fibers, nerve fiber endings, and then the detrusor muscle in 3 different layers, and that also is also quite densely innervated with all different types of nerve fibers. Just move that. So as the bladder fills with urine, the bladder wall stretches, and that accommodates then the increasing volume of urine, but that also activates efferent nerve fibers, and that contains then sensory signals that are transduced towards the central nervous system, and that provide then eventually conscious awareness of bladder fullness. And during health, bladder sensations are generally restricted to relaying the intensity of bladder fullness from these nerve fibers. But in different conditions, that can, of course, also be pain, for example. That can also be pain. Bladder fullness sensations increases as the intensity of the bladder fills, so the stretch from the bladder wall increases, and that can then be reduced or translated to different sensations that may have been discussed already, which is and that is measured then urodynamically. So first sensation of bladder filling, the first desire to void, and a strong desire to void. The nerve fibers within the bladder wall, so there's many different types of nerve fibers, and different layers are differently innervated. And as you can see here, the muscle layers respond to or the nerve fibers that are within the muscle layers respond to stretch, and they also respond to probing. There's nerve fibers that innervate both the muscle and the urothelium. Those respond both to stretch and stroke and probing. And nerve fibers within the urothelium do not respond to stretch, but they do respond to strongly to stroking and to a lesser extent to probing. So you can see that these nerve fibers are different, they respond differently, and they really relay different information towards the central nervous system. The bladder is very densely innervated, and about 80% of the nerve fibers within the bladder innervate the detrusor muscle, so the muscular layers, and they respond then directly to stretch of the bladder wall, and they also respond to transient spontaneous contraction. So during the filling phase, the bladder periodically contracts and relaxes again. These are very small contractions, and these nerve fibers also respond to those changes in bladder activity. And they have a very complex branching structure that you can also see in this image, and they are quite large and they have a complex structure. The nerve fibers in the urothelium, they're much more simple. This is then about the remaining 20%. It innervates the lamina propria and the urothelium, and we understand their function much less well, and especially than in health. I think someone might have their microphone on, and I hear myself double. I'm sure you do as well. Thank you. So, in density of those nerve fibers may show a relationship with LUTs. So different types of LUTs are related to changes in relaying the sensory information to the central nervous system. They respond too much to tactile stimuli or to pain, for example. Not tactile, but to chemical stimulation. And that can show a relationship with LUTs. And one example is inflammation. So we know that inflammatory processes also changes or induces a change in innervation of the bladder wall. And you can see that on the left-hand side here. So on the left-hand side, you can see a control group, and you can see a recurrent UTI group. And in red, you see substance P. Which is not something you need to remember, but it's used to mark for nerve fibers, and you can see that there's a very strong upregulation in the UTI group. Sorry, and then when these mice are treated with NGF neutralizing antibodies, that prevents. So that's what you see here on the bottom. That prevents sprouting, and that also alleviates the sensitivity of the bladder area, the pelvic area in these animals. And on top, you can see that this group does not show that effect. So there's a strong relationship between the innervation of the bladder wall and the clinical phenotype as well, and likely also treating the innervation or targeting the innervation of the bladder wall. That's what is shown in research like this, can help to alleviate some of the symptoms. And at the moment, the bladder has been targeted like uniformly. And we know that there are differences in innervation in the different parts of the bladder wall along those, so the bladder dome and the trigone. And in this study, which is something I did in the lab of Professor Laurie Berger in Pittsburgh, we looked specifically at the trigone of rats, and we looked at the effects of aging. And here we also saw that there was a relationship between aging. So on the left-hand side, you can see tissue from young animals. And you can see that these nerve fibers penetrate quite deeply into the urothelium, and there's quite a lot and long nerve fibers. And in aged tissue, you don't see this fine sprouting within the urothelium, and fibers overall are much more fragmented. So there's also in aging, there's a change in innervation of the bladder wall. This image shows that again. So we did immunohistochemical staining of bladder tissue of rats. We binarized all of the signal that was positive for nerve fibers. In this case, we used CGRP as a marker for nerve fibers. And in these images, you can see only the pixels that are positive for CGRP remain. And we quantify differences in those values between groups. And then we saw that the age group has significantly less CGRP positive signal. So then the magnitude of efferent nerve fibers decreases with aging in this group. So, and here we looked exclusively at the trigone, but it's quite likely, or it would be very interesting to see if there are differences between different functional areas of the bladder, or structural and functional areas of the bladder, in how this innervation changes in different conditions associated with LUTs. And we made a small start. And this was published by one of our PhD students this year. We made a small start to look at the full the full length of the bladder wall. So it's quite often been described, and then it's mostly just observational, that there are differences in innervation along the rostrocaudal axis of the bladder. But that was never quantified. And so here we did this just in healthy young mice. We stained we cut the bladder tissue along the full length. So we have all the way from dome to bladder neck. And we stained that with CGRP as well to get efferent nerve fibers. And these are mostly the nerve fibers actually that respond to, so these are C fibers. They respond to pain and to chemical stimulation, and not so much to stretch. But we see that there's an increase in those fibers towards the more caudal segments. So towards the bottom of the bladder, there's more CGRP, and this shows that there's a heterogeneity in the distribution of these fibers along the rostrocaudal axis of the bladder. And it would be very relevant and interesting, I think, to do similar things in conditions that are associated with LUTS, like aging, but also, for example, obstructed bladders, because it becomes interesting if you know like where exactly the fibers are located that are affected by a condition, and if we know like what are characteristics of these fibers, then we can target them exclusively pharmacologically, for example. So, and of course, the lower urinary tract is under direct control from the central nervous system, and these fibers, they send the sensory information that they gather from the bladder. They send this towards the central nervous system, and that's represented here in this arrow. So, and the efferent information from the lower urinary tract arrives in the central nervous system mostly at the level of the periaqueductal gray, which is this small hub here, and you can see that there's smaller sprouting towards the pontine mixturation center, and that's also called Barrington's nucleus. And I might use those two terms a little bit interchangeably, and I hope that will not make it difficult to follow. But please also feel free to interrupt me, anyone, if you have any questions at all during my presentation. And if anything is unclear, please interrupt me. So some information also arrives at the pontine mixturation center, but the PAG seems to be the main hub or relay station where information from the bladder arrives here, is sent to higher brain areas, and those higher brain areas then also send a decision whether to void or to delay voiding back towards the periaqueductal gray. From where that signal is then sent to the pontine mixturation center and ultimately back via the spinal cord back towards the bladder. And we know quite a bit about how the initiation of urination works, but we know much less about how the delay of urination works, so how exactly these brainstem areas work together to prevent the switch from storage to voiding. That we know much less about. I will go a little bit into a few studies that are underlying this model. And to start with the arrival of the efferent information from the bladder in the brain. So this is from a tracing study that shows that this information, and this is done in rats, I think. Yeah, this is done in rats. So in rats, this information arrives both in the periaqueductal gray, which you see on the right, but it also arrives in Barrington's nucleus. And that's a bit in line with what we saw in this model. But in humans, it is much more predominantly in the periaqueducal gray and to much lesser extent in Barrington's nucleus. At least that's what has been proposed. Of course, we've not been able to do the same tracing studies, but also in higher order animals such as cats, it seems to be that there's a stronger projection, much stronger projection to the periaqueducal gray rather than Barrington's nucleus. And that is also why in these animals, it has been proposed that voiding is really under voluntary control, and that the periaqueducal gray is the central hub there that regulates that and interacts with higher brain areas to facilitate that. So, and that, yeah, like I said, the role of the PAG that has been proposed by that study is also in line with how we look at it in the human. And this model or this schematic might be a bit more simple or easy to look at, but here you also see very, very well how this periaqueductal gray is really located in the very center of the model, and how the pontine micturition center, it also receives some input from the hypothalamus, but it's really very strongly under control of the periaqueductal gray. And those areas work together then to facilitate storing urine and the switch when you want it to urination. The periaqueductal gray is not one homogeneous region, even though it often seems like that. Like if you look at a model like this, it just says periaqueductal gray, but it does all of these different things. So it receives the efferent information from the bladder, it sends it to other areas, it receives information from those higher areas and communicates downstream again. That seems a lot to do for one homogeneous hub, but we know that this model should and could and should be a lot more refined. And when we look at the neuroanatomy of the periaqueductal gray, we know that it's made up into different structural columns. And for voiding, the most relevant ones are the dorsolateral column, the lateral column of the PAG, and the ventrolateral column. And for the function of the dorsolateral column, there's very limited research, but what it has been suggested to be involved with is actually the suppression of micturition, so and the delay of micturition. So this area might be related to delaying urination and facilitating the storage of voiding. The lateral PAG is the main area that receives the efferent information from the spinal cord and projects this to higher brain areas. And the ventrolateral PAG is involved in the initiation of micturition through excitatory projections to PMC. And we can already add quite a lot more, or well, it's still a little bit speculative, but there are still details that we can fill in, because as I will show later as well, the ventrolateral PAG in general seems to be the area that has the strongest connections to the pontine micturition center. So not only excitatory, but also inhibitory. And the area, the dorsolateral PAG, that has been suggested to delay voiding, and a likely mechanism to do that would be suppression or inhibition of the pontine micturition center. That area does actually not have inhibitory connections to the pontine micturition center. So if there might be connections between these areas that facilitate that. But we'll go in a little bit more here. I think I already outlined that a little bit. So ventrolateral PAG, there's very strong scientific evidence that shows that these excitatory connections, like indicated in green, there likely is a red line as well, or we know that there is a red line. I will show that later as well. And the communication between dorsolateral PAG and ventrolateral PAG, that's still purely speculative, but that might be a way that this area contributes to the delay of urination. So here you can see, so this is done with chlorotoxin B. So that's a toxin that you can use for retrograde tracing. And you can forget, of course, also the name of that tracer. But this is an injection that is done in Barrington's nucleus of the mouse. And then you can see where the neurons are located that project to that area. And this is then relatively caudal in the periaqueductal gray. So this is the cerebral aqueduct, and you can see here a border. That's a border of the PAG. And then in the ventrolateral, but also slightly in the lateral aspect of it, you can see that there are many cell bodies of neurons that project to the region where the injection was. And that's strongly in the ventrolateral PAG, but also a lot more rostrally. We can see that there are some in lateral PAG, but also a little bit in dorsolateral PAG, some neurons that project to Barrington's nucleus. But these areas, especially in mice, are very, very small, and trying to find large groups of neurons that project to Barrington's nucleus, and also to differentiate the rostral aspect from the caudal aspect, that is actually quite challenging. So this study was done in rats, and this looks at those neurons in the ventrolateral PAG. And they did with microdialysis, they measured different neurotransmitters and their presence during infusion of liquids into the bladder, of saline into the bladder in anesthetized rats. And then they see that during periods of infusion, and that infusion is equivalent in this case to frequent micturition. So if you infuse something in the bladder, then the rat also during anesthesia starts to void repeatedly. Then they see that there is a decrease of GABA within that area. So from somewhere upstream, it might be dorsolateral PAG, it might also be somewhere else. But information that arrives, or inhibitory information that arrives in the ventrolateral PAG, decreases, and that then allows neurons in that location, is the conclusion, or the proposed conclusion, that allows those neurons to excite downstream Barrington's nucleus or pontine micturition center so that the rat can void. And the opposite we see for excitatory neurotransmitters, so glutamate, that shows an increase during these periods of frequent micturition. So those are likely the neurons that we saw also within the ventrolateral PAG that have excitatory connections to Barrington's nucleus. They become more active during voiding. And stronger evidence for that is actually shown in this study, which is also, it's an enterograde tracer. So the injection was done in the PAG. And this is done in a mouse model, so VGLUT2 iris cream mice. And these mice, the virus that was used for this tracing only infects glutamatergic cells in these mice. And here you can see that the lateral PAG and ventrolateral PAG is very strong glutamatergic, so excitatory, exclusively excitatory connections to Barrington's nucleus. And that is something that we could not yet see with CTB, which is more a generic tracer. So that stains, well, glutamatergic, GABAergic, but also any other neurotransmitter, just connections in general. But when these neurons were, when they injected the optogenetic injection in ventrolateral PAG. So they injected in ventrolateral PAG, but they shone a light over the projections of those neurons in Barrington's nucleus. So then you can only activate those neurons in Barrington's nucleus that have their source, that receive their input from, or the axons of the neurons that receive their input from the ventrolateral PAG. And when those neurons are activated, then you can see that that leads to immediate voiding. So without delay, the mouse starts to void. And this was actually done in awake, freely moving mice. So that's very strong evidence that these glutamatergic PAG neurons contribute to the switch from storage to voiding. Now comes a newer part, and I will go a little bit more into methodological detail here. But when we do the same in a different mouse model, and this is then VGET iris cream mice, and in these mice you can exclusively target GABAergic cells. So we also see that there are, within that region of ventrolateral PAG, there are also many GABAergic cells. So those cells can inhibit or prevent Barrington's nucleus, pontine micturition center, from becoming active. We can see that those cells are there as well. And we would like to know what their function is. So and we can do that using chemogenetics. And we did DREADS injection, and DREADS so that's a chemogenetic method. DREADS stands for designer receptors exclusively activated by designer drugs. And by using that in this specific mouse type, we can infect only the GABAergic cells within the ventrolateral PAG, or within the injection site. So you can see here that I try to inject exclusively in the ventrolateral PAG. And on the right-hand side, you can see post-mortem that also was quite successful. And then we can see, when we also look at Barrington's nucleus, we can see that those neurons from the PAG also have their projections in Barrington's nucleus, or many of them do. And then we can use a drug to activate those receptors. So I said DRES, designer receptors, exclusively activated by designer drugs. And what that virus does, it adds a receptor to the cells that it infects. And then you can use a drug to activate those receptors. So I can make those neurons active, or I can use an antagonist and make them inactive. And then we also want to measure those animals' micturition behavior, and we can do that using a method that was designed also by the group of Hanneke Verstegen, micturition video thermography. So we have a thermal camera. We volume load mice with dextrose. So we give them an IP injection of 5% dextrose and water. And then we see what their voiding behavior is over a period of 2 hours. And you can see here, for example, this is a warm spot within the cage, and the mouse is here. So you can see 4 cages in total, actually. And here you can see a spot where a mouse just voided, and this is a void that is drying up. So we can know when the mouse voided, where it voided. And the advantage is that they often void in the same location multiple times. And if you only analyze it afterwards, so if you only look at a sheet of paper where they voided, for example, then you don't know how many voids are on top of each other. So this is quite a useful method to analyze that behavior. And we analyzed each mice 4 times, so twice with a vehicle injection. So we just inject a vehicle into the mice, just saline, and twice with a DRED agonist that activates those receptors. And by using that in VGAT mice, so where we can target the GABAergic cells, then we can take the inhibitory control from the PAG over Barrington's nucleus away. So, and the hypothesis is that if that inhibitory control is necessary for the delay of urination, then you would expect an increase in micturition frequency in the mice that have received, or when the mouse receives that DREDS agonist. Oh, I hope that makes sense. And when we run the mice with vehicles, we also see that they just behave normally. They have some voids, continent voids in the corner of their environment. They also have some defecation, some stools. But when we activate those DREDs, and so take the inhibitory control over Barrington's nucleus away, we can see that the mouse voids many times. They void they lose urine throughout the cage. So not just in the corner, but they also lose urine when they're walking. Often they actually seem to not notice that they're starting to void, so they walk a bit through their environment and then end up voiding in the corner. Ultimately, but it seems that they sort of lose that inhibitory control. So that this is very much pilot data, but that sort of shows that that they lose the inhibitory control over urination. So that could confirm also that these GABAergic neurons in the ventrolateral PAG contribute to the delay of urination. And this is all, of course, very interesting. That this is how voiding works. This is how the delay of voiding works in mice. But for patient management, it's of course very important to be able to translate all of this research to the human situation. And we are trying to unravel how the different PAG areas are interconnected with human neuroimaging, and how they are involved in load control, and that we can try to find neuroimaging human correlates of those animal mouse findings that I just showed you. And that, of course, is then a very essential step, I think, for the translation of the animal research to the human situation, and to explore any clinical potential of those methods. So we have done over the past, I think, 7 years or so, we've done quite a lot of neuroimaging work. And we have been looking a lot at the perioccipital gray. And this is really like brainstem neuroimaging is quite underexplored field. And so our first research question was very simple. We were wondering if the human periaqueductal gray can be parcellated, and that is just a word for being able to subdivide it into different functional areas using ultra-high field resting state fMRI. And then we are interested in whether there's an effect of bladder sensations on functional connectivity patterns of those parcellated PAG areas. And we collected, so we used the established algorithm. I will not go into a lot of detail, but so with fMRI research, you measure activity, oxygen consumption in small parts of the brain, and we do it in 3D areas called voxels. So it's just a three-dimensional pixel, and for us that contains information about oxygen use within the part of the brain that is represented by that voxel, and we correlate all of the voxels within the PAG with each other in their time course of oxygen use. And then we can run an algorithm over that, Louvain module detection algorithm, which is an algorithm that has been used a lot for doing this kind of thing on cortical areas. And that shows that, yes, apparently we can subdivide the periaqueductal gray into different functional areas. And that, of course, by itself is not necessarily interesting, but it's also an important question whether this organization looks biologically plausible. So we assessed the symmetry and reproducibility between empty and full bladder states. So we are trying to unravel the underlying anatomy, and the underlying anatomy should not be dependent of bladder fullness. The function likely is, but the anatomy not. And we also see that there's a very strong relationship or similarity between the empty bladder parcellation, the map, and the full bladder map. So the symmetry and reproducibility across bladder states also suggests that, yes, the clusters that we find are biologically plausible. And then we are interested in the communication between those regions and how that changes the function of bladder state. So we also used, so we had people lying in the scanner, and while we filled their bladder, we asked them what their sensations were, and we did this using questionnaires that are also used clinically on the mixed diaries that patients fill out. And questions that they're asked during normal urodynamic investigations. So we have empty bladder, that's just when we start, sub-threshold bladder filling. That is before, like, just the time before they report the first sensation. Then we have the first sensation of bladder filling. It's when the patient says, like, oh, I feel something in my bladder. I don't have to go to the bathroom yet, but I do feel that something's happening. The first desire is like, okay, I could go to the bathroom now. And strong desire is then, yeah, when they really just need to go. This is also often compared to, especially these later stages, to when you're in the car. Like, for example, the first sensation is like, oh, if I was driving home from my work, if I was going to drive home from my work, then I would go to the bathroom, because I know I can already say that in some time I will need to go. The first desire would be like when you decide to start looking for a gas station to go to the bathroom, and strong desire would really, you don't have another choice but to park your car next to the road, and you really just need, yeah, you have like an emergency strong desire to void. You need to do something about it. And then we see that between two of the clusters that we identified, there was a significant change in functional connectivity between those areas, with the highest or the strongest connectivity being around that first sensation point. So that also shows that those clusters that we identified are relevant in the context of evaluating changes in bladder sensations, right? And then we are also interested in the interaction between the periaqueducal gray and the Pontine mixturation center. So in here I show a map of the periaqueducal gray in blue, and then the Pontine mixturation center in red. So you can see how close they are to each other, also in the human brain. And we are interested in spatial similarity of the maps that we made of the PAG using that connectivity matrix of all voxels within PAG with all other voxels in the PAG. And we try to replicate that by looking at the connectivity between each of those voxels in the PAG, but now not with intrinsic data, but with data with the average signal in the Pontine micturition center. And there we see that this is just a map that we made based on that intrinsic parcellation or mapping of the PAG. And then we also make a map of correlation values of the average signal in Barrington's nucleus with each voxel in the periaqueductal gray. And then when we group those voxels in the periaqueductal gray into the strongest positive correlations, strongest negative correlations, and weakest correlations, we get this map. And visually already, this map is very similar to the map on the right. So number C is very similar to map A. And we also tested that statistically. And they also show a significant correspondence. So that shows that also based on interaction with Barrington's nucleus, like an area that we know that is strongly connected to the periaqueductal gray, we can replicate that intrinsic parcellation map. So that furthermore confirms that that subdivision that we made of the PAG, or that we can make using the Louvain parcellation algorithm, is a really useful way to start looking at subregions of the periaqueductal gray and their relationship to micturition control. And then we also looked at interaction patterns between the periaqueductal gray and the pons during filling of the bladder. And then we see that the region in the pons corresponding with the location of Barrington's nucleus shows a very strong opposing interaction pattern. And that is very complicated and vaguely termed, but we basically looked at the difference between different PAG clusters in their connectivity with Barrington's nucleus. And we see that when one cluster of the PAG, one functional region, increases in its connectivity with Barrington's nucleus, the other one decreases in its connectivity with Barrington's nucleus. And that is a bit in line with what you would expect if dorsolateral PAG is involved in suppressing micturition, and ventrolateral PAG would want to facilitate micturition. So those are opposing things that the PAG is trying to communicate to the Pontine micturition center, and which of those signals has the ultimate overhand then decides whether or not we void or if we delay voiding, right? So that we think is reflected in the connectivity changes between the Pontine micturition center and Barrington's nucleus. And I think that's what we see here. So to very shortly summarize our neuroimaging work. So can we subdivide the human periaqueductal gray? Apparently, yes. Is the organization biologically plausible? Yes. The symmetry and reproducibility suggest that it is biologically possible. Does the communication change between those regions with bladder state? Yes, it does. And we can detect interaction with the Pontine micturition circuitry and also relate that to changes in bladder fullness. So that's what we have been able to do with the neuroimaging work so far. And we're also exploring changes or differences between healthy people and people with an overactive bladder. And what sacral neuromodulation can do there. And this is also all really pilot work. We've published a bit. Unfortunately, did not include that. I can see if it's in my hidden slides. No, it's not. Differences between healthy people and overactive bladder patients. We can see that there is also the consistency between these maps, between an empty bladder and a full bladder, is stronger in overactive bladder patients than it is in healthy controls. So in healthy controls, there's more variability in the mapping over time. And in overactive bladder patients, there is very little variability. And actually in the few, I think maybe 2 or 3 sacral neuromodulation patients that we've included so far or analyzed so far, we can see actually that these were successful patients that responded to the treatment. There we see that they also start to show more variability in the mapping of the periaqueductal gray over time as the bladder fills than they did before the treatment. So here we can really see we can try to understand how these therapies work, which is something that we don't really know yet. And we can try, or hopefully that will give us useful information about which patients are likely to respond to that therapy and which patients we can use with that therapy. And to illustrate that a little bit more, I would like to close with this. So if we can study the system, and then I mean the bladder itself, so we know a lot more about the innervation of the bladder, how that relates with different types of lower urinary tract dysfunction and different types of neurogenic conditions, and how the brain helps to control the lower urinary tract, both with voiding, with urination, and what happens in different patient populations. So we know a lot more mechanistic detail, and that might help us to change how we think about patients. So if you look at these 4 patients that I put here, like they all have the same symptom, but they are 4 different patients. So you can have peripheral hypersensitivity, spinal hyperexcitability, reduced cortical control, or altered bladder wall structure and compliance that all result in urgency, patients that experience urgency. So they have different biology, the same symptom, but also likely a different optimal treatment. And I think it will be useful, and the basic science in this field will hopefully help us to switch or to move from the current label, which is rather general, neurogenic detrusor overactivity, to labels that are more based on mechanisms and also identify the likely target, right? So we often, at the moment, we describe what the bladder is doing, like neurogenic detrusor overactivity, but that does not really tell us why that is happening. So the same urodynamic phenotype may result from different underlying mechanisms. So, for example, altered efferent signaling, spinal hyperexcitability, or impaired supraspinal control, so really something happening in the brain. And if we can combine advances in our understanding of the mechanisms that are associated with these different conditions and integrate that with the diagnostic tools that are already available, so we know how these patients differ in their urodynamics, for example, but also other available diagnostic tools, we may be able to classify patients more precisely, and that then really would help to move from symptom-based treatments to more targeted and mechanism-based treatment options. So that was my presentation. I would be very happy to take any questions that you might have.
Thank you, Professor. It was very good.
Yeah,
so
thank
do
you.
have any... Yes, Professor.
Oh, I don't want to interrupt you, Abhinav. I think
No.
I also want to thank Rick. It's a very, very interesting talk and everything very new for me. Although I'm such a long time already neuro-urologist, of course, I knew some aspects, but it's very interesting, especially, can you go back to your last slide? Or this, no, to the 4 patients.
Yeah.
Yeah. So this would, of course, so in the future, you would suggest not discriminating neurogenic and non-neurogenic detrusor overactivity, but rather what are the mechanisms? And that would give us a different look. Is that?
Yeah, I think so. I think within neurogenic, of course, there's many different patients that would show the same, that would show neurogenic overactivity based on
but also in the
experience.
non-neurogenic.
Yeah.
I think in the non-neurogenic OIB, we definitely don't know anything. With the neurogenic, we think we know something, because something has to do with the neurogenic on the assumption, in spinal cord injured patients, it's easy. But in patients with diabetes, for example, it could be neurogenic and non-neurogenic, and we basically don't discriminate now. We only say he has a neurogenic disease, so it could be a neurogenic detrusor overactivity. And I think it really is a great. It opened a whole new world of targeting, and especially, and then of course in the future, hopefully also altering our approach. Thank you.
Yeah, thank you. Yeah, of course, that also very much holds up for a non-neurogenic detrusor overactivity.
Can you show the next slide again?
Yeah.
Can you show your next slide again? See, here you says in this slide you still said neurogenic detrusor overactivity. Actually, to be precise, you should here write detrusor overactivity, right?
Yeah, it's just an example of a current label that we use that I think.
But
detrusor
actually,
overactivity, yeah.